So this is a real public-private success story that we're going to relate. ATRACTion Project, Autoimmunity Information by RNA Sequence Single Cell Analysis for Therapeutic Innovation. This is an acronym, very difficult acronym, but we can understand already what it's going to be about. And to talk about this ATRACTion program, we've got three top-ranking scientists and specialists of the situation which have gathered together to get this project on the stage. Frédéric RIEUX-LAUCAT, you're from Inserm, Descartes-Sorbonne Paris-Cité University, and one of the pillars of Imagine Institute. Mohammad AFSHAR, you are CEO of Ariana Pharma, and Michel Didier, you're project leader at Sanofi, big pharma, French big pharma, very well known. And the three of you have worked on this ATRACTion project, which shows the connection between public and private interests. And I let you the floor to explain and give us the most important issues of this project.
Okay, thank you very much. Good afternoon. So I will give you a brief overview of the project, and then we will start the discussion.
Of course. And just before, I just remind you, don't hesitate to ask your questions during the presentation. Thank you.
Well, so if we can have the first slide. ATRACTion, yes, it's autoimmunity and inflammation through RNAseq analysis at single cell level. And so today I'm with Mohammad AFSHAR and Michel Didier, respectively from Ariana Pharma and Sanofi, and we are going to discuss this project that is supported by the RIEUX-LAUCAT call. So, in fact, the whole project starts from primary immune deficiencies and patients that are affected by autoimmunity and inflammation. And the unmet clinical needs are the precision diagnosis and adapted specific treatments for these patients. So they are rare diseases, but in fact, over the planet, there are 500,000 people that are affected by primary immune deficiencies. 2/3 of them are less than 19 years of age, so it's a pediatric disease. And 1/4 of these patients are developing autoimmunity and inflammation. So we have a unique cohort at the Necker Hospital of more than 5,000 patients recorded in the reference center. And our concept is to develop a multi-omics analysis based on single cell analysis and omics. So the objective is that starting from this large group of patients, what we observed in the last years is that one gene defect may lead to very several presentations. So if we take the example of CTLA4 or STAT3 gain of function mutations, they can lead to autoimmune enteropathy or autoimmune cytopenia. And we recently described the SOX1 deficiency in patients that have lupus, psoriasis, autoimmune cytopenia. So very different clinical presentation with the very same defect. But we may have one phenotype with several genes, and that is the case, for instance, for the Evans syndrome, so autoimmune cytopenia. And we described that there are more than 20 genes that may explain this phenotype. So this is leading to diagnosis and treatment wandering, because one patient will come to different departments. And so these patients are treated according to the clinical signs, and then an unpredictable proportion of these patients will not respond to the treatment. And our hypothesis is that this is due to the molecular heterogeneity of these patients. So the goal of this project is to solve this problem and this diagnosis and treatment wandering. And we will process 250 patients and 250 controls through different single-cell omics analysis in order to re-stratify the patients according to the signatures that will be developed by these approaches. And by network inference, we plan to characterize the molecules that could be the most adapted for these patients. So in this project, we will define new software of prognosis and therapeutics, validations of biomarkers, an atlas of molecular interactions that is leading to autoimmunity or inflammation, and of course a personalized treatment of these patients with most probably repurposed compounds. So, in summary, the goal of this project is to develop kits and tools to diagnose these patients. So, these undiagnosed patients will go to a clinician in the hospital. We will collect the clinical sign, and then using the diagnostic kits that will be developed, and an online algorithm, similarity algorithm, we will place the patients according to the archetypal patients cohort that define the signatures. That should allow to know what are the affected pathways, what are the possible treatments, and possibly what's the ongoing clinical sign that the patients may develop. So overall, it's a tools for guidance in diagnosis and therapeutics decisions that are made by the clinicians in primary immunodeficiencies with autoimmunity, and probably that can apply to more common diseases in adults. So just to give you the overall work packages of this project. So in the first work package is the cohort of patients. So as I said, 250 patients and 250 healthy controls. That will be analyzed through the different omics, single cells, as well as with the metabolomics and lipidomics and metagenomics, in order to see if there is an impact of the microbiota on the onset of the disease or the variable clinical expression of the disease. All the partners are listed in front of the different work packages. And that will lead to pathological cell clusters, signatures, and using the technology of artificial intelligence and network inference, we would like to make all these omics talking together in order to develop biomarkers, transcriptomic signatures, molecular pathways, and that will be validated, and drug screening that will be validated in a clinical trial on a subgroup of patients. So that is the view of the project. And just to show you some preliminary data, on the next slide, if I can move. Yes, here is an example of single cell analysis. So it's a single layer of omics performed in 4 patients with idiopathic juvenile arthritis. And what we could see here is that we could see a pathological cluster of cells that is only present or detectable in the patients. And therefore, we can analyze the differentially expressed genes in this particular cluster that will lead to now validations by the surface of the cells or the functions of the cells. So, in conclusion, it's a new approach to decipher the molecular basis of pathophysiological mechanisms and for adapted treatment accordingly to these pathways that are affected. The strength of the project is based on the unique cohort of patients, and also to the fact that each omic layer may provide key information. And the challenges are the handling of this very big amount of data, and with the help of Ariana Pharma to connect these different types of data to talk together and see whether we can improve the diagnosis and therefore the therapeutic decision that could be made for these patients. So no, I think that's the whole project, and the discussion is open with my partners to.
Yeah, of course. You just said that Mohammad AFSHAR and your company Ariana Pharma is very important in this ecosystem, and to get the data and to the data and the connection with the clinic. So how do you work? Tell us.
So I think just to follow on what was just Frédéric was just mentioning, I think when you talk about multi-omic data and the challenge of making things talk to each other, I think one just needs to realize just the sheer complexity of that, and just in simple numbers. If you're looking at the genomic analysis, you may have you look at a few mutations on a gene panel, and you're looking at 10, maybe a few hundred genes. here we're talking about looking at the all the genes that are expressed at a single cell level, and then adding into that the gut microbiota, etc. So suddenly, per patient, we're talking about maybe 400, 500,000 items that need to be manipulated. And Frédéric mentioned that the goal is to have 500 patients. So it means that for each patient you have 500,000 elements that describe them, and you need to manage their combinations and link that to potential biomarkers, potential markers that define certain type of specific disease or certain response to a particular drug. So the challenge that we have is to use the right AI methods to be able to combine all this data. And clearly, because we have what's called these very wide matrices, so relatively small number of samples, but extremely well defined, we can't use just statistics. And that's where all the machine learning tools come in. And that is really the focus of Ariana as a company.
This is your specialty. What you
Absolutely,
can do.
absolutely, and really, it's translating these complex multi-omic data into specific, again, biomarkers, specifically of response to a therapy, or biomarkers that can help as surrogate endpoints to follow the evolution of a particular disease, or to help us identify novel indications for existing drugs, etc.
We'll go back to Ariana Pharma, but Michel, Didier, what's your connection and what's the interest of a big pharma like Sanofi in this project?
So our interest for us, is really at the target validation and screening, So as you know, the change we are facing most of the time in the pharma industry is to really put a finger on the right target, the target for which we believe that if we find some kind of modality to regulate the target, we have high impact on the disease. So clearly, what is really interesting in this strategy, we are starting with what we call human mutant, in which we know that we have specific mutation in genes that are going to drive a lot of change and pathogenic changes, in the immune system. So starting already with this kind of cohort, in which we can really launch high resolution analysis of the system is really important. And what I will say also in terms of target validation, and in the first work package in single cell, it will be also we will contribute to put some additional data to the analysis of the patient cells. For example, we will also launch some single cell analysis, but by running what we call perturbation analysis. That means we will take normal cells, we will also stimulate here and there, specific pathway that are being demonstrated to be involved also in the disease. So we will contribute to also produce some data. And what we are saying here is really critical, because the way we are going to produce the data will be pretty similar to the data that will be generated from the patient. And we believe it will be really helpful to construct, a more consolidated database and to help, in the analysis, So that is the first level, And from that, later on, when we do a more integrated analysis, what we hope, and especially Frédéric was indicating, the inference network, it means that we will generate some model of immune cells regulation, inter-immune cell regulation. And for that, we believe we can identify here and there, some key driver, again, on the deregulation of the immune system. And that will produce some really interesting target candidate that we will further, validate, in models, including phenotypic models. And then our main contribution will be screening, will be to use that models either to launch some phenotypic screening, or eventually also, so it could be screening with small molecule, but it could be also to pre-identify some target for which we believe if we have, if we co-target, this particular driver, we may have some effect, and so we can also imagine to derive some biologics, modality and so on. So we will be, our work package is more, in screening, identifying some tool compound, identifying some modalities that could further restore the immune dysfunction in these particular models.
Okay, and you're going to focus on rare or specific disease, but you maybe want to find some clues for more common disease.
Exactly, and that is also one of the goals is really to use this rare disease. Of course, what is the key priority is to find some therapeutic for this rare disease. But we believe that by identifying some biomarker, we can further, better re-stratify more common disease that could be much more heterogeneous, and to identify some a cohort of patients in which we believe that target could be engaged, and increasing the chance of success of testing, some molecules that could be really beneficial.
So this is very ambitious, and we understood that even if there are only 250 patients involved, it's a huge volume of data and a huge capacity to study them and to find some clues and to reach the purpose you have. We have many questions for you. It proves that your project is very attractive. The first question by Alexander Belot. How distinct monogenic disease will be included, and how much different gene defect among the 250 patients? And is this level of diversity and heterogeneity is an issue for the analysis? Who wants to answer? Yeah, Frédéric.
Well, thank you, Alexandre, and good afternoon.
Yeah.
Well, in fact, I mean, it's a very good point, because as you may know, there are more than 420 different monogenic diseases defining the whole PID population. So we will first focus on the most common ones in order to have models, and then, depending on the inclusion of the patients, we will go in maybe more rare patients. And one of the goals is to include idiopathic juvenile arthritis patients, because it's a pathology that is quite common, both in children and in adults. There are few genetic causes. I mean, I think there is maybe one at the moment, and so that is the strength of this project, that we can go beyond the genetic cause, because if we can see the dysregulated pathway, regardless of the gene defect, and that may give us some trail for the gene defect, but then we can go directly to the targets and see how we can manipulate them. So and then to answer the question, we have roughly 20 types of genetic defects we would like to include, but that will depend. We have 4 years to do that, and that will depend of the will of the families and the inclusion of the patients. So it will be step by step that we will see how are the.
Yeah, because it's just starting now.
Right. We just got the authorization from the regulatory, the CPP. So now we can start.
Within a month, 2 months, to get, for example, the cohort gathered and.
Well, the cohort of 250, it's planned to make it within 3 years.
Okay. Step by step.
Yeah,
Okay.
step by step, because so we need the blood samples, we need the feces and the urines.
Okay.
So it's a kind of in terms of logistics, it's quite heavy.
But first patients very soon.
First patients very soon.
Okay, and they're all connected to Necker.
They are all patients from one center at the moment, but of course we let the door open if there are the possibilities to include patients from outside Necker. That they will be welcome. We just have to make some regulatory adjustment.
All right. Another question, how to distinguish the causes or just effects from your results? The best way is by AI. This is what we talked about, yeah?
Yeah.
Mohammad?
Yes, I mean that's a great question, and I think that the causality is one of the issues that people have been talking about a lot, and there are methods that try to identify associations and then characterize the direction of the association, and this is something that we're using. Again, that is really important, especially if even with this huge amount of data, you may be actually interested on 2 patients that bear a particular combination of markers that are associated to a particular outcome. So the whole challenge is to be able to identify these, specifically with your AI, and then be able to provide that causality hypothesis with the associated characteristic numbers, etc., to the group that can then assess it, not only in the context of the data from the clinical trial, which is ongoing, but also in the context of the general knowledge in the field. So not everything that we know about these diseases is included in the patients that we're screening. Hence the importance of having what's called explainable AI, so that you can explicit why you found an association and how you can interact with the rest of the knowledge.
You won't have what we call the black box.
Absolutely. I think it's critical here.
Yeah.
I think once you go to the next stages of, when you're in the discovery phase, it's really essential, again, to have a, completely white box and having an explainable AI that addresses exactly this question.
Another question for you, because it's just curiosity. This is technical or logistic. You will have a huge volume of data, but you have to be close to the clinics, let's say, to the cohort and Necker. How will you manage that? I remember that Amazon, for example, has trucks to be close to the centers. How will you manage that?
Absolutely. And I think that, I think once you get, again, I think what's fascinating about this project is that when you start looking at the details of it, then you can see also the complexity of the logistics at different levels. At a simple level, as you mentioned, accessing the data, this kind of data, once it's generated, cannot be just shoved over the internet and transferred. Which means that we're working with the various teams that are involved, including the teams at the Institut Imagine, to actually get the calculations close to the data. So you can't, so initially for the first samples, etc., the data can be transferred and we can create all our prototypes. But at the end, all the calculations need to be at the closest to the data.
So computers, not only
absolutely
data, but computers.
Computers, but again, that means that you need significant amount of calculation, CPU power, etc., to handle these several 100,000 descriptors that are combined. So there are some logistic challenges on the computing side, which is also exciting. Absolutely, absolutely, yeah.
It's very exciting, and good luck. And this is another question from Mehdi Deliud, but I think we have already answered. I'm not sure. What is the source of the data you need to make a good use of the AI tools?
So I think Frédéric has addressed that. I think we're getting incredibly good quality data, which is homogeneous within the clinical trial, which is planned here. I think that is a unique opportunity to have this. I think this data can then be also augmented by additional sources of our real-world evidence data that could come. But I think I think one of the opportunities here is to have quality data, which then potentially will lead to quality results from the consortium.
Right. Go back to the, I'd say, big science. How much single-cell analysis is representative of a complex tissue composition that can evolve with time and during therapy? A question by Hanan.
It's a difficult question, of course.
Indeed.
In fact, that is also a very important. It's a key point, because in the project, what is planned is that we have a first inclusion, and then the samples at the inclusions, and for the patient and the patient only. There will be a sample 1 year, in the follow-up of the patients within 6 months to 1 year, and that will be used as a backup and as a sample to validate what we have seen on the first inclusion. And of course, the most difficult question is to see the impact of the treatment, and then to characterize what will be the dysregulation related to the disease, and what will be the consequences of the treatment. So that is not really the goal of the project. The project is really to try to include patients as much as possible before they get heavily treated. and that will be also key. And that is why this project is a one-center project, at least at the beginning, because we want to have all the information regarding the treatments. We try to avoid antibiotics in the patients in order to not modify too much the microbiota, and so there are a lot of restrictions that are really required to have a clean set of data, both at the in the clinical aspects and at the molecular aspects.
Is there a specific profile for your patient's age or sex or?
No, it's well. They must be. They are pediatric patients, so they are under 18.
Under 18.
Otherwise, there is no.
This is the only constraint.
Yes, autoimmunity or inflammation.
Okay.
To complete, I mean, the Frédéric answer is also, that's why also profiling and single cell compound responses are really important. So it's not going to replace, profiling patient under treatment for sure. But we will also run some single cell analysis again with in vitro stimulated, cells by different pathway with and without, a reference, treatment. We have been testing that in the past. It can provide some additional signature how the cells are responding not only to the pathway but also, to the treatment. So that it will be also something that would be useful to build up, the database and to provide some kind of reference signature. Again, it will be in acute condition, but it can, in some situation, it can be really helpful.
Okay, so we just said that first patients could be involved within 1 or 2 months, but what are next steps from now? What do you hope to succeed in the future years from now?
Well, first we will have to generate the different data. So there are single-cell RNAseq, there is ATRACTion, there are proteomics, and then all the metabolomics, lipidomics, and metagenomics. So I think we will have a first view of the first analysis by the end of the year for each layers of omics. But then we'll see how long it takes to try to make them speaking together. And that is the challenge. And today it's difficult to say when we will be able to say yes, these data are talking together and they provide further information and they help.
This is your job.
That's right. That's right. So I think for us, there are really 3 challenges here. One is just on the technical side, as we mentioned, just the heavy duty part.
Yeah.
I think the second challenge is making all these things talk and extracting the biomarkers. And finally, I think one of the goals of the project is to develop a therapeutic decision support software at the end. This is something that we had done previously in oncology. And again, here is an opportunity to learn this new field in this context, and really develop over the duration of the project, this type of software that could be then used routinely. That's a challenge.
Yeah, there is a lot of deep learning involvement in your.
Well, actually, at this case,
strategy?
it's not. It's really what we're trying to do is really at this stage is to do almost the opposite of deep learning.
Okay.
Because deep learning works extremely well when you have a very large number of examples.
Oh, that's right.
And so that at the end, the deep learning tells you, it just recognizes a particular object without telling you why. Here we're at the first stage. What's really important is the why. So if the system identifies a patient that may respond to a drug, for example, we want to know why.
Why? Okay. Because several times we don't really know sometimes why they do respond and why they don't respond. So
yes.
This is exactly what you want to target. Okay, this is really great. Thank you very much for participating and presenting your ATRACTion project. So Frédéric RIEUX-LAUCAT from Inserm and Imagine Institute, Mohammad AFSHAR from Ariana Pharma, and Michel Didier from Sanofi. Thanks to the 3 of you. Thank you very much.